So good morning, everyone. Thank you for tuning in to my presentation. My presentation is titled The Association of Speech and Language Deficits with Duarte Galactosemia. This was conducted with Dr. Duarte at Boston Children's Hospital, whose team I've been a part of for the past 3 years now, and I'm very fortunate to have this experience. So a brief introduction to galactosemia. What is it? It is the inability to process galactose. And galactose is a simple sugar, which is a component of lactose commonly found in all dairy products and some legumes like chickpeas. And because the body has an inability to metabolize galactose, things like breast milk can become poison to infants. So when you knock out one step in this pathway of metabolizing galactose, the upstream products begin to accumulate, and that causes a lot of severe outcomes in the newborn period. Fortunately, this is detected by newborn screening in the United States and Europe. Chances are everyone in this room was screened for it at birth. And it's also important to note that there are several biochemical and clinical variants of this disease. So the two main types of galactosemia that are seen in the world are classic galactosemia and Duarte galactosemia. Classic galactosemia is much more rare at 1 in 40 to 60,000, but the clinical outcomes are much more severe. So like I said, breast milk is poison to infants in the newborn period. And if galactosemia isn't diagnosed in the short term, it usually results in death within 1 week of life. Luckily, the only known treatment is a galactose-restricted diet, so simply avoid all forms of galactose in the diet, in which case the acute symptoms are delayed until later in development, at which point we see cognitive impairment, motor impairment, speech and language deficits, cataracts. 80% of women with classic galactosemia have primary ovarian insufficiency. And the enzyme in classic galactosemia has 0% activity, so completely non-functional. Duarte galactosemia, on the other hand, which is what this project focused on, is much more common, 1 in 3,500 globally, and there's no treatment recommended, so it's not recommended that the children adhere to a galactose-restricted diet. There's no real severe long-term outcomes, and this has led researchers to question, is it even a disease? Because it doesn't require treatment, it doesn't have any severe outcomes. Why do we care about it? So what's the problem? I just said that there aren't any real reasons to care about this disease. Well, it's because there's a lack of consistent long-term profile. So all of the prior research that is focused on Duarte galactosemia has had really heterogeneous results in terms of flip-flopping between there are no cognitive differences whether or not you adhere to a diet, and then there are cognitive differences and speech deficits. There are delayed socio-emotional development and reduced auditory processing. And then the most recent study is back to no differences. And each of these studies has their own set of strengths and weaknesses, but it really highlights how elusive some of these long-term outcomes are. And the type of long-term outcomes that we do see are very similar to those that we see in classical galactosemia, albeit a little milder. So we're trying to really establish what the link is between galactose metabolism and speech and language development, which is what we commonly see most in the study clinic that we were at. And the reason why this link is so elusive is because these two pathways are seemingly independent of each other. So the hypothesis that we had was the GALT locus, so the gene responsible for galactosemia, is associated with the genes responsible for speech and language development, so that there's some type of association or link between the two that's explaining these outcomes. So the research methods that were employed were, number one, a comprehensive literature review, which used a kind of hub and spoke model with the three main categories being the history of galactosemia and the body of knowledge surrounding research that has already been done, the body of knowledge surrounding speech and language deficits, and the intersection of the two, so how galactosemia and speech and language deficits are related to each other. Number two was a clinical chart review conducted at Boston Children's Hospital. And number three, it was a meta-analysis of genome-wide association studies, also known as GWAS. And a meta-analysis is really analyzing all studies that have been previously conducted that have involved the genes of interest. So the results of the literature review showed that pathogenesis of speech and language deficits, so how these deficits seen in clinic and how they are formed are not well studied in general. The leading hypothesis, which is what we've had since the 1980s actually, is that galactose exposure in the fetal brain damages speech and language centers, but there's been no real proof to really conclude that this is true. Second, networks of seemingly unrelated proteins can directly lead to a disease state, so these complex interactions can lead to some type of clinical outcome, and that India has an unbelievable prevalence of the allele responsible for Duarte galactosemia. It's on the tune of about 18% of the population, and it's completely not well studied at all. So the clinical chart review was comprised of all patients with classic or Duarte galactosemia seen at the Boston Children's Hospital Metabolism Clinic from 1978 to 2013. We measured whether or not they had participated in early intervention services or special education services. And we also administered two tests that are well established in the field called the Bailey tests and the WISC, which are performed on separate age ranges depending on what's most appropriate. And they lead to several scores, such as the cognitive score, the language score, and the motor score, which have a standardized value mean of 100, which is average, plus or minus 15 for standard deviation. And the study was IRB approved. So what we found in the clinical chart review is that both Duarte galactosemia and classic galactosemia participate in special services at much higher rates compared to the general population. And most notably, 32% of the Duarte galactosemia individuals in our study participated in some type of speech therapy, compared to the classic at 71, which is much higher, but still it is severe considering the overall population's participation rate is only 3%. So it's clear that there is elevated participation rates in these special services among Duarte galactosemia and classic galactosemia. Moving on to the Bailey scores, we saw relative weaknesses in the motor category compared to the cognitive and language. And this was true for both Duarte and classic. But what was important was the differences between the other two groups seemed to get better with age. So before 1 year, the motor score was significantly weaker than the other two categories. But as the children aged, the motor score improved so that it really was not a relative weakness anymore. And this was again true for the Duarte and classic groups. So what we saw or concluded from the clinical chart review is that the Duarte group has some key similarities between the classic galactosemia group, and that there are these transient complications in childhood that resolve with age, but it is still something worthy of being studied. So the GWAS studies used major online open source tools to analyze the genomes to see what genes are possibly associated with each other. And this is a sample output from some of my analyses. The blue triangle at the top represents a mutation that's responsible for Duarte galactosemia, and all the other triangles below that represent mutations throughout the genome that are highly associated with it. So on the y-axis is the R² value, and the color coding is yellow, orange, red, going up in R² value, the red being an R² value of 0.8, which would mean it's pretty highly associated with the Duarte galactosemia mutation. And we were only looking at 500 kilobases of genomic space. The whole human genome is on the tune of 4.8 billion base pairs. So this represents a small fraction of what we could analyze, and we were limited by computational capacity. So in total, we identified 150 mutations in the nearby genomic space, which are highly associated with the allele responsible for Duarte galactosemia. This boils down to 14 genes and many more unknown transcripts that we don't know what their function is. And we're only looking within 500 base pairs, so, like I said. So moving on, broad conclusions is that a literature review was conducted, and there's a severe lack of information on long-term outcomes, but the Indian subcontinent has an extremely high potential for further analysis. The clinical chart review concluded that individuals with Duarte galactosemia may experience transient speech and language deficits. That certainly warrants further analysis as well. And the GWAS meta-analysis found several gene and regulatory targets for subsequent analysis. And I'd just like to thank the following people and institutions for their help with this work, and I'm happy to take any questions.